Hi friends, today we are going to see the past year NEET questions for the chapter Respiration in Plants, and today in this video we will cover part 3. So earlier we have completed 2 parts, so those who haven't gone through, kindly go through. Okay, so let's begin with the first question here. This question was asked in the year 2017. Okay, which statement is wrong for Krebs cycle? Okay, so you're given 4 statements here, but before we go into the solution, let us discuss what is Krebs cycle. What is the other name for Krebs cycle? It is also called as citric acid cycle, or you can call it as a TCA cycle. You have many names for it. It is a series of chemical reactions that is used by all aerobic organisms to release the stored energy through the oxidation of acetyl-CoA, and that is derived from carbohydrates. Energy what you use or the system produces, or what the cycle produces, is derived from carbohydrates, fats, proteins, and into ATP and carbon dioxide. So what is liberated in the last is the ATP and the carbon dioxide. Okay, so this is just an introduction that I gave you for TCA cycle. Now we'll go one option by option. There is one point in the cycle where FAD plus is reduced to FADH2. No, actually that is wrong. During conversion of succinyl-CoA to succinic acid, a molecule of GTP is synthesized. Okay. Option C, the cycle starts with the condensation of acetyl group, that is the acetyl-CoA with pyruvic acid to yield citric acid. So option C is the right answer. As we already saw, the citric acid is also known as tricarboxylic acid cycle, or the Krebs cycle, or TCA cycle, or citric acid cycle. It is a series. What did we saw? It is a series of chemical reactions, right? It's a series of chemical reactions. What else we saw? It will take place in aerobic organisms or in the presence of oxygen. Apart from that, the organisms generate energy through the oxidation of this main ingredient, which is called as acetyl-CoA. Apart from that, the rest of the options are correct. So here, if you see, the cycle starts with condensation of acetyl-CoA with pyruvic acid to yield citric acid is wrong. Okay, so here they are asking us which statement is wrong according to the Krebs cycle. Krebs cycle begins with the condensation of acetyl-CoA with oxaloacetic acid to form citric acid. It will not take place with pyruvic acid but oxaloacetic acid. Therefore, the correct answer is option C. So here you have the TCA cycle. This is how it happens. You have the acetyl-CoA which gets converted into citrate into NAD. Then you have the FADH2, NADH, all that. Okay. So this is a very easy pathway actually. So which statement is wrong here? If you see, it is option C. Next question here. This question was asked in the year 2016. Specialized epidermal cells surrounding the guard cells are called as dash. What is epidermal cell? Can we just discuss about it? What is epidermal cells? Epidermal cells, so you must have heard the skin. When we talk about skin, we all know the epidermis is the outermost layer of the 3 layers that make up a skin, right? The epidermis layer provides a barrier. What does it do? It provides a barrier to infection from environmental pathogens and regulates the amount of water that is released from the body into the atmosphere through a trans-epidermal water loss. Okay, so you're given 4 options here. What do you mean by complementary cells? We will go one by one. Then only you will understand the answer. Complementary cells. Complementary cells is a mass of cells in plants that are formed from the cock of cambium at the position of lenticels. It is a group of loosely arranged cells that aid in gaseous exchange through cock. So complementary cells are associated with plants and they are also called as mass of cells. So it is not associated with the epidermis. So complementary is not the answer. Before going into subsidiary, what do you mean by bulliform cells? What do you mean by bulliform cells? See, bulliform cells are actually large bubble-shaped cells. Okay. They are bubble-shaped epidermal cells that occur in groups on the upper surface of the leaves and especially found in monocots. Okay, so specialized epidermal cell that surrounds the guard cells, what is they asking here? So this will not surround guard cells, therefore this is not the answer. Now what do you mean by lenticels? We saw in the last question, I mean the last option that we saw, complementary, right? It is one of the many raised pores in the stem of the woody plant that allows gaseous exchange to happen. So once again it is not lenticels. Now what is the answer? The answer is subsidiary cells. Okay, now what is this subsidiary cells? A plant epidermal cell that is located next to the guard cell. Okay, so subsidiary cell is only present next to the guard cell. Okay, next to guard cell in the stoma of the leaf. So where is it present? In the stoma of leaf. Okay, and differs in structure from the other epidermal cells and also called as an accessory cell. Therefore the correct answer to this question is option B. Okay, next question here, which was asked in the year 2008. The hemiosmotic coupling hypothesis of oxidative phosphorylation proposes that ATP is formed because of dash. Okay, now I will show you a picture which shows what is hemiosmotic hypothesis. See, during the photophosphorylation steps, when the electron carrier molecule passes on to its electron to the electron carrier on the inner side of the membrane, protons are released into the inner side of the lumen of the membrane, and this is called as hemiosmotic hypothesis. Before that, tell me what you mean by hypothesis. What is the general meaning of hypothesis? See, hypothesis can be described as maybe I can give you an example here. It can also be called as a proposition made on the basis for reasoning without any assumption of its truth. That is, it's a saying. Okay, it's an explanation that is made on basis of limited advances or evidences. So that is the basic meaning of hypothesis here. So I have clearly mentioned what is hemiosmotic hypothesis. Now you're given 4 options here, right? High energy bonds are formed from in mitochondrial proteins. ADP is pumped out. So what do you think will be the answer? The correct answer is a proton gradient forms across the inner membrane. So what do you mean by proton gradient now? What is gradient first? See, proton gradient can be explained or it can be defined as an electrochemical gradient also. It is a gradient which happens with the help of electrochemical potential, usually for an ion that can move across the membrane. So you all know we are all blessed with electrons and protons. All that is called as ions, right? The gradient consists of 2 parts, the chemical gradient or the difference in the solute concentration across a membrane, and the electric gradient. So what are the 2 gradients that you can see? Electrical and chemical gradient. Okay, always remember this. And the ions that are involved in it are called as the ion gradient. So here the proton gradient forms across the inner membrane. So here if you see clearly in the picture, they clearly tell you what actually the electron carriers, the electron which passes. So it is related to the proton gradient forms across the inner membrane. Okay, the correct answer is option C. Next question here. This question was asked in the year 2007. Okay, all enzymes of TCA cycle are located in the mitochondrial matrix except one which is located in the inner mitochondrial membrane in eukaryotes and in cytosol. Cytosol is nothing but the cytoplasm in prokaryotes. This enzyme is what? Which enzyme is it? So here you have 4 enzymes given here, right? You have isocitrate, you have malate, you have lactate. They are asking us about the enzyme that is present in the inner mitochondrial membrane. Okay, now each enzyme has a different function. Okay, but the correct answer is succinate dehydrogenase. Succinate dehydrogenase or succinate ubiquitin oxyreductase, also known in mitochondria as a complex 2, provides a link between the citric acid cycle and the membrane bound electron transport system. Apart from that, what you have to know about succinate dehydrogenase is that succinate dehydrogenase plays a very important role in TCA cycle. Okay, it is also abbreviated as SDH. Okay, and this is actually a coenzyme. So what is it? It's a coenzyme. Okay, and this is actually a respiratory complex too, and it is an enzyme complex that is found in many bacterial cells and in inner mitochondrial membrane of eukaryotes. It is also the only enzyme that participates both in citric acid cycle and also in electron transport chain. Apart from that, if you see, the rest of the enzymes will be in the outer part itself, will be located in the mitochondrial matrix itself. The one which is present inside is succinate dehydrogenase. So that is what they have explained to your right side, if you clearly see. Therefore, the correct answer is option C. Always remember two things here. This is the only enzyme that can participate in both citric acid cycle and electron transport chain. Okay, next question here, which was asked in the year 2007. The overall goal of glycolysis, Krebs cycle and electron transport system is the formation of dash. I think this is a very easy question for you to answer, right? So here, if you see, they're asking us, we all know any cycle or any process that takes place in our body will synthesize ATP. They are all metabolic pathways that happen in a living cell, which provides ATP, and that is the energy currency of any living cell. So here you have two things here. So it is normally not sugars or nucleic acids. B and C is not the right answer. But look at A and D. ATP in one large oxidation reaction and ATP in small stepwise units. If you clearly watch glycolysis, Krebs cycle or any cycle, the synthesis of ATP is not an overall process, or it is not a sudden process. It happens in stepwise, and in each step you have some ATP to be stored and it will collect, and in the final step you'll have a good amount of ATP. So therefore ATP is in small stepwise units. If you clearly watch or if you clearly see the cycle, you will understand. You'll have, if you see in the glycolysis, the first step will have 2 ATP, the next step will have 3 ATP. So it's a small step. The storage is in the form of small units. They're not happening all of a sudden. Okay, so therefore the correct answer to this question is option D. Glycolysis, Krebs cycle and electron transport system, they all give up ATP, which is the energy currency of any living cell. Glycolysis takes place in the cytoplasm, while Krebs cycle also takes place in cytoplasm, while ETS, which is called as electron transport systems, will take place in mitochondria. Therefore the correct answer to this question is option D. Next question here, which was asked in the year 2005. Okay, Hemiosmotic theory of ATP synthesis in the chloroplast and mitochondria is based upon dash. So this is once again an easy question for you because we earlier discussed about it, right? Whenever we talk about chemiosmotic theory or hypothesis, we always talk about the ions. We have two gradients, one is the chemical gradient and the electric gradient. And when we talk about osmotic theory, chemiosmotic hypothesis or theory, they predominantly talk about proton gradient. Therefore, the correct answer to this question is option D. We already discussed in the earlier question, right? In the beginning. So here you have the electrochemical proton gradient, which is, I've just given you an example, a picture to represent that. Okay, apart from that, what else you can say about this proton gradient is, what else you can say? See, proton gradient, or you can call it as electrochemical gradient, the gradient will consist of two parts. Okay, will consist of two parts. So here what happens is the ions will be usually transferred, or it can move across the membrane. What does it do? The ions keep moving across the membrane, and when it is needed, they will be used up. So this is what it does. So those two parts resemble the chemical and the electrical gradient. Okay, next question here, which was again asked in the year 2005. During which stage in the complete oxidation of glucose are the greatest number of ATP molecules obtained from ADP? Okay, you're given 4 options here. Okay, you're given 4 options here. Now, what do you mean by, ATP, you know what is the full form, right? It is adenosine triphosphate. Now what is the what can you say about the electron transport chain, ETS pathway? How can you describe it? It is already called as ETC. It is a series of complexes. What does it do? What does it do? It transfers electrons, right? It transfers electrons from the electron donors to the electron acceptors via the redox reaction, which involves both reduction and oxidation, which will occur simultaneously. And this will couple this electron transfer with the transfer of protons across the membrane. So already we saw about this, where we spoke about the proton gradient during the Hemiosmotic hypothesis or theory, where we said we have certain ions which keep moving across the membrane, and when it is required for any cycle or any process, they will be obtained and used up for the process. Likewise, the three main steps involved in the transport chain is that aerobic respiration occurs in 3 stages. When you talk about electron transport chain, it includes glycolysis, the Krebs cycle, and the electron transport. In glycolysis, glucose will split up into 2 molecules of pyruvate, and this will result in net gain of 2 ATP molecules. So this is how it occurs. So here, during which stage a complete oxidation of glucose will attain a great number of ATP molecule to form ADP is in the electron transport chain. So far I've explained you what is electron transport chain, why is it important, and why is ions important. Okay, next question here. During anaerobic digestion of organic waste, such as in producing biogas, which one of the following is left undegraded? Okay, so look at this question here. We are talking about anaerobic, that is in the absence of oxygen. Okay, so here you have cellulose, lipids, hemicellulose, lignin. Okay, cellulose is obviously not associated with the organic waste. Hemicellulose, lipids, once again not associated. The correct answer is lignin. Lignin is a complex polymer of polypropane units which are cross-linked to each other with a variety of different chemical bonds. Other than that, if you want to know anything about lignin, it can be described as see, it is during anaerobic digestion, it will not be able to get degraded. Main thing is that. Okay, lignin are particularly important in the formation of cell walls. So what are they included? They are important in the formation of cell walls, especially in wood, okay, bark, because they are good in rigidity. Okay, apart from that, lignin fills a space in the cell walls between cellulose, hemicellulose, and pectine components, especially in vascular and support tissues, xylem, tracheids, and apart from that, this is the activity. It will fill the spaces so that the cell wall becomes more rigid, and thereby helps in the survival of the organism. Okay, lignin is a natural, sometimes it is also used as a natural glue. Okay, it is used as a natural glue that holds wood fibers together. It is renewable and abundant, and some of it is already been sold for pulp mills. So I've given you a brief introduction about this lignin. Therefore, the correct answer to this question is option C, lignin. Okay, next question here. It's just a kind of reaction that they have shown here. Which one of the following concerns photophosphorylation? Now, before going into the equations given here, what do you mean by photophosphorylation? How can you describe it? See, photophosphorylation happens it's a process which happens among the photosynthesis, right? In the process of photosynthesis, the phosphorylation of ADP to form ATP. So what happens here? ADP to form ATP using energy of sunlight is called as photophosphorylation. So here photo tells you it is light, right? Photo means light. We all know that. So what is the process happening here? It is a phosphorylation of ADP to ATP. Cyclic photophosphorylation occurs in both aerobic and anaerobic conditions. Only 2 sources of energy are available to living organisms. One is sunlight that we naturally get, and the other is the reduction oxidation reactions which take place, which gives us energy. Okay, So this is an introduction that I've given you. Phosphorylation also refers to the use of light energy from photosynthesis to ultimately provide energy to convert ADP to ATP. Thus, this is a replenishing of the universal energy currency in living things. Now, phosphorylation refers to the addition of phosphate in the presence of light. So, which process has correctly added phosphate to it? Which option do you think will be right? So, look at option A. AMP plus inorganic phosphate gives you ATP. That is also happening in the presence of light, but we all know it is ADP. Therefore, option C is the right answer. ADP plus inorganic phosphate gives you ATP. Okay, therefore the correct answer is option C, because we just now saw that conversion of ADP. Okay, that conversion of ADP to ATP requires light, and phosphorylation is addition of phosphate. So this correctly manages. Option C correctly tells you it is ADP plus inorganic phosphate in the presence of light gives you ATP. Okay, next question here. In which one of the following do the two names refer to one and the same thing? Okay, this is again an easy question for you. You have tricarboxylic acid cycle and urea cycle. It is wrong, because TCA cycle is also called as Krebs cycle. It is not called as urea cycle. Look at option B, Krebs cycle and Calvin cycle. Both are different cycles. Look at option C, tricarboxylic acid cycle and citric acid cycle. So option C is right. So they've given you the other names of each cycle. You just have to match and tell. Now, for example, if you take option A, it should see both the options should talk about a single cycle. Now, if you take TCA cycle and urea cycle of option A, both are not referring to the same cycle. They are referring to two different cycles. Therefore, the correct answer will come for option C, where they refer to one cycle there. TCA is also called as citric acid cycle, Krebs cycle. So option C is the right answer. Very easy question for you to answer, right? Okay. Next question here, which was asked in the year 1997. The mechanism of ATP formation both in chloroplast and mitochondria is explained with help of what? Okay, you're given four options here, right? When we talk about the ATP formation both in chloroplast and in mitochondria, what do you think will explain that? See, you're given four options here: relay pump theory, Munch's pressure. So we'll go one by one. That will be better, because directly going into the answer might not help you. What you mean by relay pump theory? Okay, relay pump theory can be explained as a process, and this was actually proposed by Goldswick. Proposed this clambering, or this is also called as relay pump theory, to explain the vertical movement of water through the plant. He thought that the rhythmic change in the water potential of living cells, especially xylem and phloem, especially xylem, because xylem is involved in the transport of water. So he spoke about that particular theory. So the theory is all about the you can talk about the vertical movement of water. So therefore it doesn't talk anything about the ATP formation. So A is not the answer. Now coming on to Munch's pressure. What do you mean by Munch's pressure or Munch theory? Okay, so here if you see this theory behind mass flow hypothesis, which is called as pressure flow hypothesis. What is it otherwise called as? Pressure flow hypothesis. Okay, and this describes the movement of the sap via the phloem. So this is a movement of sap. Basically we saw about relay pump, movement of water, and this is a movement of sap. Therefore the correct answer to this question is Hemiosmotic theory which was proposed by Mitchell. According to this theory, the energy liberated during the electron transport chain performs the osmotic work of accumulating H+ ions, therefore conserve energy in the building of the proton gradient. So which correctly talks about ATP formation is option C, Hemiosmotic theory of Mitchell. Okay, therefore the correct answer is option C. Now you have one more option left. Did you ever think about that? What do you think will be the answer for Hollande-Wentz model? Actually C is silent there. You can call it as Hollande Wendt's model or Wendt theory. So this is a theory which states that the plant trophism in roots and shoots are due to the unequal distribution of the growth regulator, which is called as auxin, in response to light or gravity. So this Wendt's theory or model also talks about the they talk about the root and the shoot system. So which correctly talks about ATP formation is option C, Hemiosmotic theory by Mitchell. Okay, next question here. This question was asked in the year 1997. Okay. In Krebs cycle, FAD participates as electron acceptor during the conversion of dash. Okay. See, in the conversion of the succinyl-CoA, what we are taking here? We are taking succinyl-CoA to succinic acid. This will get converted into succinic acid. We all know this process. Energy is liberated during hydrolysis of succinyl-CoA is used in the synthesis of GTP. For the conversion of the alpha-geta glutarate to succinyl-CoA, NAD plus and CoA are required. What is CoA? It's an enzyme. Conversion of the fumaric acid to malic acid involves simple hydration. Then you require water. FAD participates in conversion of succinic acid to fumaric acid. Therefore, the correct answer to this question is option C. Succinic acid will get converted into fumaric acid. Okay, the correct answer is option C. Next question here, which was asked in the year 1990, repeated in 92 and again 97. Connecting link. Okay, they're talking about the connecting link between glycolysis and Krebs cycle is, or before entering Krebs cycle, pyruvate is changed to dash. See, we all know the end product of glycolysis is pyruvate, right? But what actually connects that link, if you see, it is acetyl-CoA, right? We all know that. What gives that link between glycolysis and Krebs cycle is that acetyl-CoA. Okay. It enters the mitochondria and it is oxidatively decarboxylated to acetyl-CoA before entering into the Krebs cycle. Therefore, the correct answer to this question is acetyl-CoA. Here they are asking us not the end product, but they are asking us the connecting link. So kindly read the question carefully, because we tend to go wrong in these questions because they sound familiar to us. So always read it clearly and then answer. Next question here. So this question, oxidative phosphorylation involves simultaneous oxidation and phosphorylation to finally form what? Yeah, this is again an easy question for you. We all know any process, any cycle that we talk about, the ultimate aim is the synthesis of ATP, which is the end product, and which is actually used by the living cell in the form of energy consumption. Now this oxidative phosphorylation is the synthesis of energy rich ATP from ADP, an inorganic phosphate that is related to the oxidation of reduced. You have the help of other coenzymes which help in synthesis of this ATP from ADP. So oxidative phosphorylation, which involves simultaneously both, they will give you an end product which is to finally form ATP. Easy question, right? Next question, Krebs cycle occurs in dash. Very easy question for you to answer, right? We all know Krebs cycle occurs in mitochondria, also called the powerhouse of the cell. Glycolysis, if you see, it will occur in the cytoplasm of the cell, also called as cytosol. So this question is very easy for you to answer. Krebs cycle occurs in mitochondria. You have the picture of mitochondria to your right side. I think this is the most easiest question we have seen so far, right? So this question was asked in the year 1996. Fine. So with this, we complete part 3 of respiration in plants. Hope to meet you in the next part of the same chapter. Thank you for watching my video.